Chapter 14, which is the last chapter for your exam 4, has to do with Mendel and the gene idea. The main concepts covered in this chapter that we'll be going over are that Mendel used a scientific approach to identify 2 laws of inheritance. Inheritance patterns are often more complex than predicted by simple Mendelian inheritance, and many traits follow Mendelian patterns of inheritance. So who was Mendel? Mendel is considered the father of genetics. He was an Austrian monk. He worked at a monastery, then he went to university for a few years where he took physics and chemistry courses, and then he went back to the monastery and taught there and other places. He did his famous research in 1857, and that is when he began breeding garden pea plants. That was the organism that he did his research with. He created a big garden in his monastery where he conducted thousands and thousands of crosses. And he came up with the particulate hypothesis. And this is the idea that parents pass on discrete heritable units to their offspring. Now today we call these genes. Back then we didn't have a word for it, so they used discrete heritable units. So basically what this is saying is that traits don't blend together. So if something is blue and something is yellow and passed down, you don't get something green, for example. Now, we see sometimes this can happen, but this isn't normally what happens. Now, when we talk about a heritable feature that varies among individuals, this is what we call a character. So flower color, for example, would be a character. Each variance for a character is called a trait. So for example, purple versus white. So we have the character, which could be flower color, and the different variants or varieties of it, perhaps could be purple and white. Now, why did Mendel choose to work with garden pea plants? Well, there's a couple of different reasons. One is that they were available to him in many different varieties. So he actually studied 7 different characters, where he could have 2 versions of each, and we're going to look at what some of those were. Another reason why he used pea plants is that they had a short generation time. It didn't take long for them to produce seeds. They also produced large numbers of offspring, so they produced a large number of seeds. And you'll see later, if you take upper level biology or statistics courses, that usually when we do experiments, we want large numbers. That makes our results more accurate. And another reason he used pea plants was that matings could be controlled. He did either self-pollination or cross-pollination. Now, pea plants, their flowers can produce both pollen, which releases sperm, and eggs. So they naturally self-pollinate themselves. He sometimes did manipulations where he could cross-pollinate them. So these were easy to work with. Now again, Mendel looked at characters that occurred in 2 distinct alternative forms. Now he started with varieties that were what we call true breeding. These were organisms that produce offspring of the same variety when they self-pollinate. So for example, something that's tall always produces an offspring that's tall if it self-pollinates. If we have a plant that's short, if it self-pollinates, it always produces a plant that's short. That's what true breeding means. When Mendel mated individuals, he did it for 2 contrasting true breeding varieties. So for example, a true breeding tall and a true breeding short. The true breeding parents are what we call the P generation, or P stands for parental. The hybrid offspring of the P generation are called the F1 generation. F stands for filial, so the first filial generation. When F1 individuals either self or cross-pollinate with other F1 hybrids, we call that the F2 generation. And we'll be looking at what a hybrid is in a few slides. Now, when Mendel crossed true breeding white and purple flowered pea plants, all of the offspring ended up being purple. You can see that in this first slide. Here's our P generation. a true breeding purple with a true breeding white. All of the offspring were hybrids, but they appeared purple in color. And we're going to see that what a hybrid means is that it has an allele from each parent, and those two alleles are different. Now, when Mendel crossed the F1 hybrids, he found that many of the F2 individuals were purple, but some were white. And it ends up that the ratio was roughly a 3 purple to 1 white ratio. So there's a 3-to-1 ratio in the F2 generation. Now, Mendel called the purple flower color a dominant trait and the white flower color a recessive trait. And it's important to note that the factor for white flowers was not diluted or destroyed, because when we got to the F2 generation, we did see some white flowers. So it was still there, it just wasn't showing itself in that F1 generation. Now, this pattern of inheritance was observed by Mendel in 6 other characters in the pea plant. And when we use the term, again, heritable factor, that's what we now call a gene. Here we see the different characters that Mendel investigated. So there was flower color, purple versus white. And you see in the F2 generation, we got roughly a 3-to-1 ratio. Seed color, yellow versus green. Seed shape, round versus wrinkled. Pod color, green versus yellow. Pod shape, inflated versus constricted. Flower position, axial versus terminal. And stem length, tall versus dwarf. And again, the important thing to note here is that one trait was dominant, one was recessive. And in the F2 generation, there was roughly a 3 dominant to 1 recessive ratio. Now, I'd like you to know 1 or 2 of these characters, but you don't need to memorize all of the different characters and what those traits were. This is just to illustrate the point of what Mendel was doing. Mendel developed a hypothesis to explain the 3-to-1 inheritance pattern that he observed in the F2 offspring. And there's 4 parts to the development of this hypothesis. The first is that there are alternative versions of genes that account for variations in inherited characters. These are called alleles. And again, for example, with flower color, there's a purple allele and there's a white allele. And this is just showing this picture on the bottom. If you look at the blue and the red, those are the 2 chromosomes, say 2 number 5s, for example. And it's showing the location for the flower color gene. It's showing in one it's carrying a purple allele, and in one it carries a white allele. And as long as it has 1 copy of the dominant allele, it produces that dominant color. So that's the first step. That's the first thing he figured out, that there are different versions of genes. These different versions are called alleles. Step 2, for each character, an organism inherits 2 alleles, one from the mother and one from the father. So there's 1 from each parent. The 2 alleles at a particular locus or location on the chromosome may be identical or they may differ. If the 2 alleles at a locus differ, the dominant allele will determine the appearance. The recessive allele has no noticeable effect. And from these findings, Mendel came up with the law of segregation. And this states that the 2 alleles for a heritable character separate during gamete formation and end up in different gametes. So an egg or a sperm gets only one of the 2 alleles that are present in the organism. This slide goes over some basic genetic vocabulary. So if an organism has 2 identical alleles for a character, we call it a homozygote. This organism is true breeding. Remember, we talked about that a few slides ago. An organism with 2 different alleles for a gene is what we call a heterozygote, hetero-different. We use a capital letter to represent a dominant allele and a lowercase letter to represent a recessive allele. The term phenotype refers to the outward appearance. So it could be purple, it could be white, tall, dwarf, round, wrinkled. Those are all phenotypes. A genotype is the actual genetic makeup, so the actual alleles. Now, if an individual, and we'll look at the example with purple and white, is homozygous dominant, capital B, capital P, or if it's heterozygous, capital P, lowercase p, they have the same phenotype. They both appear purple, but they have different genotypes because one has 2 dominant alleles, the other has 1 dominant allele and 1 recessive allele. And remember that we said in general, the dominant allele is going to be the one that shows up. There are a few exceptions, but this is a different type of inheritance pattern. So if it has 2 dominant alleles, or if it's heterozygous, it's going to show the dominant trait. The possible combinations of sperm and egg can be shown using a Punnett square. This slide indicates the difference between phenotype and genotype. So on the left we have phenotypes. We have purple and white. If we look at that F2 generation, we have on average 3 purple for every white. So the ratio for phenotype would be 3 purple to 1 white. If we look at genotype, we can get a purple from a big P, from a big P, a little p, big P, and a little p. Now, big P, and little p, big P are exactly the same. They put them in the same order there. So we have 3 combinations that can give us purple and 1 that can give us white. Now, if we're looking at a genotypic ratio, we have 1 big P to 2 big P to 1 little p. So again, phenotype is the appearance and genotype is the actual genetic makeup, the actual alleles. This slide is illustrating Mendel's law of segregation. So here we have the purple generation. We're starting with 2 true breeding individuals. One has purple flowers, so its genotype is capital P, and one is true breeding for white flowers, so its genotype is lowercase p. The purple flower can only make gametes that have a capital P because that's all that it has for that trait. Similarly, a white flower can only produce gametes that have a lowercase p because that's the only version it has of that trait. When these 2 combined in the F1 generation, all of the offspring are going to be big P, little p. It gets 1 allele from one parent and 1 allele from the other parent. So the genotype of the F1 generation are all going to be big P, little p. The phenotype of the F1 generation is going to be all purple because the dominant trait is going to show, the recessive trait is hidden. Now, when 2 F1 individuals mate, we have a difference here than we had up top. Half of the gametes that these flowers produce will have the capital P allele passed down. The other half of the gametes will have a lowercase p passed down. So in the F2 generation, this is showing how we would do a Punnett square. So for a Punnett square, we set up rows and columns that correspond to the different alleles. So if we're doing a single trait cross where there's 2 alleles, we're going to make 4 boxes. On the top of the columns, we'll put what could be passed down from one parent. So again, we have 2 parents that are big P, little p. So one parent can pass down a big P, so that's the head of one column, or a little p, that's the head of the other column. The other parent, so let's say the female we're putting before the rows, it can pass down a big P or a little p. So whatever alleles can be passed down, we put one gender on the top and one gender on the sides. And they have sperm on the top and eggs on the left-hand side. You could reverse that. You could put the eggs up top and the sperm on the side. It doesn't matter which side you put them on. And then what you do, you just combine the different alleles to see your different possibilities. So in the first box, for the column, we have a big P, and for the row, we have a big P. So one possibility is a big P individual. In the next box, we have a lowercase p from the sperm and a capital P from the egg. So this individual would be big P, little p for its genotype. It would be purple for its phenotype. In the bottom left-hand corner, the sperm passes down to big P, and the egg passes down to little p. So this also produces a heterozygote offspring, a big P, little p, whose phenotype would be purple. And then in the final box, we see there's a lowercase p passed down from the sperm, and then a lowercase p passed over from the egg. That individual is lowercase p, which is the phenotype of white. So if we look at this Punnett square in totality, out of every 4 offspring that are produced, 3 will be purple and 1 will be white. Now, Mendel derived the law of segregation by following a single character. So for in the last example, he was looking at the character of flower color. The F1 offspring produced in this cross were what they call monohybrids. That means it was heterozygous. Something that's a hybrid is a mixture. So it's heterozygous for one character. Mono 1, hybrid a mixture. So 1 capital P, 1 lowercase p. Now if you cross 2 monohybrids, we also call this a monohybrid cross. Now Mendel had another law of inheritance that he came up with. And he came up with this law by following 2 characters at the same time. And he found that by crossing 2 true breeding parents that differed in 2 characters produced a dihybrid individual. Now a dihybrid individual is an individual that's heterozygous for 2 characters. So maybe flower color and plant height. So it's like a monohybrid, only we're doing it with 2 traits instead of just 1. If you cross 2 of these individuals, that's called a dihybrid cross. Now by looking at 2 characters, he came up with the law of independent assortment. And this states that each pair of alleles segregates independently of any other pair of alleles during gamete formation. So basically, what he's saying in the example of, say, flower color and plant height, that where certain alleles go for plant height doesn't affect where the alleles for flower color are going to go. They're independent of each other. Now, this holds for 2 different situations. So, for example, if the genes are on different non-homologous chromosomes, this will hold. So, for example, if the flower color gene is on chromosome 1 and the flower height gene is on chromosome 2, they're going to assort independently. Where one goes doesn't affect where the other goes. It also holds if these 2 genes are on the same chromosome, but they're very far apart. So one's, say, on the upper end of the chromosome and one's on the lower end. They also tend to assort independently. Because if you remember during meiosis, we have crossing over, so the material often gets mixed up. Now, when this doesn't hold true, if genes are located near each other on the same chromosome, if they're near each other, they tend to be inherited together. Now, sometimes inheritance of characters by a single gene may deviate from simple Mendelian patterns, such as in the following situations. When alleles are not completely dominant or recessive, when a gene has more than 2 alleles, or when a gene produces multiple phenotypes. So we're going to see there's actually different degrees of dominance. So often we have complete dominance, and that's what we've talked about so far. When the phenotype of the heterozygote and the dominant homozygote are identical. So the purple flowers, whether it has 2 purple alleles or if it has a purple and white allele, appears purple. That would be complete dominance. Sometimes we have what's called incomplete dominance. This is when an F1 hybrid has a phenotype that's in between the 2 parents. And this is what we see on the right-hand side. Here in the P generation, we have a red individual, it's homozygous for red, and an individual that's homozygous for white. And when we look at the F1 generation, we see that the offspring, which has one red allele and one white allele, appears pink. So this is an example of incomplete dominance. We could also have what's called codominance. And this is when two dominant alleles affect phenotype in separate ways. So two alleles are being expressed at the same time. So we see this when we look at the ABO blood grouping. We see that different antigens are present, and this is codominance. Now, dominant alleles are not necessarily more common in populations than recessive alleles. So one baby out of 400 in the United States is born with extra fingers or toes. This condition is called polydactyly, and it's caused by a dominant allele, but it's found much less frequently in the population than the recessive allele. This trait is also found in cats, and this is an example of a cat, and if you look, this cat has 6 toes on each paw, which is more than it's supposed to. Again, we see this in cats, we also see this in humans. If any of you have heard of the author Hemingway, at his estate there's still a whole colony of cats that have polydactyly. They have extra toes. Now, most genes exist in populations in more than 2 allelic forms. We've been talking about 2 alleles, dominant or recessive. Oftentimes, there's multiple alleles, more than 2. One example are the 4 phenotypes of the ABO blood group in humans. So you can have type A blood, type B blood, type AB, or type O. And this is also showing, like I mentioned before, codominance. So figure 14.11 is showing the multiple alleles that are involved in the ABO blood grouping. Instead of the 2 alleles, like we've been talking about, there's 3 alleles. One allele is IA. This is a dominant allele. Another allele is IB. This is also a dominant allele. And then we have a lowercase I. This is a recessive allele. IA codes for an enzyme that attaches carbohydrate A. IB codes for an enzyme that attaches carbohydrate B. Allele lowercase I doesn't code for the attachment of any carbohydrate. And when we say attached, these are going to be attached to red blood cells. So we have 3 different alleles, IA, IB, and lowercase I. So with these 3 alleles, there are 4 possible different phenotypes, A, B, AB, and O. If an individual has 2 copies of A, or if it has a copy of A and the recessive I, it's a type A blood. If it has 2 copies of B, or 1 copy of B and a copy of lowercase I, it's type B blood. If it's type AB, it has 1 copy of the A allele and 1 copy of the B allele. And we could also, again, see here codominance. It's going to be attaching both carbohydrates, A and B, to the red blood cell surface. And then phenotype O, blood group O, the genotype is if it has 2 lowercase I alleles. It doesn't attach any surface carbohydrates. Most genes have multiple phenotypic effects. This is something we call pleiotropy. For example, pleiotropic alleles are responsible for multiple symptoms of certain hereditary diseases, such as cystic fibrosis and sickle cell disease, which we'll be talking about a little bit later on. Quantitative characters are those that vary in the population along a continuum. So, for example, height. Quantitative variation usually indicates polygenic inheritance. This is when there's an additive effect of 2 or more genes on a single phenotype. So some examples of polygenic inheritance. 1 is height. Now, in determining height, there's actually over 180 different genes that play some role. Some are more important than others. Now, these aren't alleles of the same gene. These are actually different genes that play a role. We also see polygenic inheritance involved in skin color. We also see it involved in several different diseases. So, for example, heart disease, alcoholism, mental illnesses. We often see multiple genes are involved. Now, sometimes the phenotype for a character depends on the environment as well as the genotype. Traits that depend on multiple genes combined with environmental influences are called multifactorial. They depend on both the genes and the environment, multiple factors. So in A and B, we see hydrangeas, and they vary in color depending upon what type of soil they're grown in. So if they're grown in basic soil, they appear pink in color. If the same genetic variety is grown in acidic soil with free aluminum, they appear blue in color. So the environment is influencing the color. Now, humans are not good subjects for genetic research like garden pea plants are. They have a long generation time, so it's 9 months before an offspring is actually born. Parents produce relatively few offspring, often 1, occasionally 2, but you don't have a large number. It's also not ethical. With humans, we use what are called pedigrees. This is a family tree that describes the interrelationships of parents and children across generations. You can trace inheritance patterns of traits and predict future offspring with these. And here we have a listing of some common symbols used in human pedigrees. And there's a few of these I want you to know. So a circle is a female and a square is a male. If the circle or square has a line through it, that individual is deceased. If the circle or square is blank on the inside, that individual is unaffected. If the circle or square is completely filled in, that individual is affected. And if the circle or square is halfway filled in, they are a heterozygote. The other symbols, I'm not interested with you knowing what those are for now. Now this slide shows a human pedigree for cystic fibrosis. So you should be able to read this. So what we see here are 2 parents, individuals 1-1 is a male that's heterozygous, and a female, individual 1-2 that's heterozygous. Those 2 individuals have 3 children. The first of the 3 is 2-2. This is a normal female. Then there's individual 2-3. This is a male that has cystic fibrosis. And then we have 2-4. This is a female that's heterozygous. So she's normal in terms of her phenotype, but she's carrying that recessive allele. Individual 2-2 is married to individual 2-1. They have 3 children. 2 of those are female, 1 is male. They are all normal. Daughter 2-4 is married to male 2-5. He is a heterozygote like her. They have 4 children. The first is an affected male. Next is a normal female. Next is a normal male. And finally, at 3-7 is an affected female. Many genetic disorders are inherited in a recessive manner. That means that only those that are homozygous for the recessive allele show it. They have to have 2 copies of the recessive allele. Some individuals we call carriers. These are individuals that are heterozygous. So they carry the recessive allele, but they are phenotypically normal because they have 1 normal allele. Now, in this example, we're looking at an example for albinism, so having white hair. On the right-hand side, you can see there's 2 children. The one on the right is an albino. There are 2 parents. In the Punnett square, we're going to do to predict what the probabilities for the different offspring are. both parents appear normal but are heterozygous. So they each carry that recessive allele that can lead to albinism. So the sperm can either pass down a normal allele or the recessive allele for albinism, and the same thing for the egg. So out of every 4 offspring, 3 will appear normal and 1 will be albino. In terms of genotypes, 1 will be totally normal, 2 will be carriers, they'll be heterozygous, and 1 again will have the 2 recessive alleles for albinism. Now before we mention cystic fibrosis is the most common lethal genetic disease in the United States. 1 out of every 2,500 people of European descent have cystic fibrosis. This disease is inherited in a recessive manner, so you need 2 recessive alleles to have the disease. The recessive alleles result in defective or absent chloride transport channels and plasma membranes, which basically means there's going to be a buildup of chloride ions outside of the cell because there's no transport channel for them to go through. Remember we said that some diseases can show multiple different effects on a phenotype. And I said we get to that later. These are showing some of the multiple effects that cystic fibrosis can have. Mucus buildup in some internal organs, and abnormal absorption of nutrients in the small intestine. Those are 2 of the symptoms of cystic fibrosis. Another inherited disease is sickle cell disease. Now, sickle cell disease is recessively inherited and affects about 1 out of 400 African Americans. It is caused by the substitution of a single amino acid in hemoglobin. And again, hemoglobin is found in red blood cells. Now, in homozygous recessive individuals, all the hemoglobin is abnormal. It has a sickle shape, which is why it's called sickle cell disease, and it has a tendency to clump together. Some symptoms of these individuals include physical weakness, pain, and organ damage. Heterozygous individuals are also called sickle cell trait carriers. They're usually healthy, but they can show some symptoms. About 1 out of 10 African Americans has sickle cell trait. This is just showing the differences between the red blood cells. On the left-hand side is what a normal red blood cell looks like. On the right-hand side is what a sickled red blood cell looks like. And again, you can see it has an obvious sickle shape to it. And that makes it easier for them to clump together and to get stuck and block the blood vessels. The last 2 diseases we talked about, cystic fibrosis and sickle cell, are both recessive inherited disorders. Now we're going to look at an example of a dominantly inherited disorder. Now, sometimes dominant alleles can cause lethality and cause a disease that's lethal. These are rare and arise by mutation. But again, sometimes they just cause a disease itself, not necessarily death. One example is achondroplasia. This is a form of dwarfism caused by a rare dominant allele. And you can see that in the individual on the right-hand side. And here we have 2 individuals that mate in our little Punnett square. We have an individual that's a dwarf. He has 1 capital D and 1 lowercase d that he can pass down. And the D is what's actually causing the dwarfism. It's a dominant allele. And on the left-hand side, we see there is a normal mother. She has 2 recessive alleles for the dwarfism, lowercase d. When you do this Punnett square, you'll see that 2 out of every 4 individuals will be dwarf, and 2 out of every 4 individuals will be normal. Sometimes the timing of onset of a disease significantly affects its inheritance. So one example is Huntington's disease. This is inherited in a dominant fashion, and it is a degenerative disease of the nervous system. It actually has no obvious phenotypic effects until the individual is about 35 to 40 years of age. Once the deterioration of the nervous system begins, the condition is irreversible and fatal. So a lot of times people have children and they may have Huntington's disease and not know it because it doesn't show up until an individual is about 35 to 40 years of age. Now in this slide we're seeing the Manx cat. These are two of my cats. They're brother and sister. One was taken when they were kittens, one is older. And you can see that they have a very shortened or reduced tail. So Manx is caused by mutation in a dominant trait that affects the spine. The mutation shortens the tail. So if you have one copy of the dominant allele, the cat will have a shortened tail. You can see the little boy on the right has a stub, and the girl on the left, she has a very even smaller nub. If an individual has two dominant copies, it's a homozygote, then the condition is lethal and the individual isn't even born. It dies as an embryo. Genetic counselors provide information to prospective parents concerned about a family history for a specific disease. They can use pedigrees to help determine the risk a couple's child will have a certain disease. For a growing number of diseases, there's also testing available that can identify carriers and help define the odds more accurately. On this slide, we see two methods of fetal testing that can be done. One is amniocentesis. This can be done at the 15th week of pregnancy, and this is when the liquid that bathes the fetus, the amniotic fluid, is removed and tested. On the right-hand side, we see chorionic villus sampling, abbreviated CVS. This is done at the 10th week, and a sample of the placenta is removed and tested. Now, a positive of the one on the right-hand side, the chorionic villus sampling, is that it can be done earlier in the pregnancy. It can be done 10 weeks, whereas the amniocentesis is usually done at 15 weeks. One of the negatives, however, is that chorionic villus sampling has a slightly higher chance of a miscarriage. So you have to weigh your options when determining whether you want to have one done over the other. Some genetic disorders can be detected at birth, so a newborn can be screened. And these are simple tests that are routinely performed in most hospitals in the United States for some disorders. One common test is for PKU. This is a recessively inherited disorder that occurs in one of every 10 to 15,000 births in the United States. So if a person has two recessive copies of allele, they lack an enzyme that can break down phenylalanine. And this could cause mental impairment later on. So if a newborn is screened and they find out that that newborn has two recessive alleles, they can feed them a diet that does not have phenylalanine in it. That individual will then develop normally. So screening is a very important tool that can be used.